When a SAK or sexual assault kit is examined at the Houston Forensic Science Center, a number of initial tests allow lab technicians to retrieve evidence originally gathered by the emergency room staff or specially trained forensic nurses, also known as sexual assault nurse examiners, or SANE. When the kit arrives at the lab, a forensic scientist opens the kit and removes the evidence. The kit contents are expected to match the contents indicated in the hospital report. The analyst breaks the seals, opens the kit, and organizes its contents. Lab personnel always wear face masks to avoid contaminating evidence. DNA is sensitive, and so it is not recommended to talk or sneeze while collecting the evidence. If a nurse should contaminate the evidence with their own DNA, the forensic scientist in the lab would have no way of knowing it was a nurse rather than the suspect's DNA. If a nurse suspects they have contaminated a sample, they should inform the lab. The lab may request a DNA sample from the nurse. To avoid such contamination, a mask can be worn during the evidence collection process. Paperwork is generated, and the lab adds the case number and the item numbers to the items contained within the kit, so each item is uniquely identified. Swab packaging should be clearly marked to indicate the origin of the evidence. Vaginal swabs can be grouped together in the same package. The analyst then counts and documents the swabs. A microcentrifuge tube is labeled with the assigned case number and the item number. The swab tops are portioned, so part of them can be placed into a tube for analysis, and another portion can be saved should future testing be needed. When collecting swab evidence, roll the swab for even distribution. This ensures the entire swab is covered in evidence and helps ensure the analyst does not take an empty portion of the swab for testing. The tube is loaded onto a QIAcube. This instrument uses automation to differentially extract DNA from the swab. Differential extraction separates and extracts sperm DNA from non-sperm DNA. The vial is loaded in several more machines that will quantify the amount of human DNA in a sample, create millions of specific copies through amplification, and finally separate the DNA fragments with a genetic analyzer. Special DNA analysis software is used to interpret the DNA results for evidence samples from the kit to known sources of DNA. Here, the forensics DNA analyst can determine if the item of evidence and the known source of DNA have the same origin. At this point, eligible DNA profiles can be entered into the National DNA Database, also known as CODIS, and connections can be made between evidence in a case and a convicted offender, or between evidence in two different cases. Undergarments require a different type of processing to remove DNA evidence. The analyst will first have to remove the undergarments from the evidence bag. The panties shown here are tagged with a label that includes the case number and the item number. The undergarment is recorded on the evidence inventory form and photographed. Once photographed, the evidence needs to be examined using an alternative light source. The Omnichrome machine at the Houston Forensic Science Center helps an analyst find possible semen stains. Once the semen is located, it fluoresces under the alternate light source. That area is cut out for testing. When packaging sexual assault kits, it is important to wear gloves and a face mask at all times. This will prevent DNA evidence from being contaminated with the nurse's DNA. Sometimes hair also needs to be covered. Always examine the sexual assault evidence collection kit to ensure it's not been damaged or tampered with. Label the exterior of the sexual assault kit with the patient's name, sex, age, hospital location, date of examination, attending examiner's name and signature, the law enforcement agency, and the case number. Alternatively, the hospital label can be used for identification purposes. The chain of custody information should be completed correctly and appropriately. Envelopes from the SAK box should be labeled with the patient name, date, and exact time of collection, the nurse's initials or signature, and, in some cases, the reason for the collection. For example, when using the debris collection envelope, it is useful for nurses to be clear about the potential evidence. For example, is this semen or contact DNA? For some labs, the analysis method will differ if the semen is potentially present. A helpful note will also originate with the victim and could be something such as, "He licked my neck." Cross out items not being collected, such as vaginal smear or penile swabs for a female patient. If a sample has been collected, indicate that by checking the appropriate box at the bottom of the envelope. Use of kit contents are based on a patient history and assessment, which means not all items within a kit will always be used. Unused envelopes should be left blank or simply not returned to the box. Order of collection should be determined by the history and assessment of the patient, with the exception of cases where sample loss is a possibility. In instances of an oral assault, the evidentiary oral swabs should be collected prior to the known saliva swabs. Before swabbing, check the proper collection box on the container and then assemble the swab carton box. When collecting a swab, roll the swab to ensure even distribution of DNA. This ensures the entire swab is covered in evidence and helps guarantee that the analyst does not take an empty portion of the swab for testing. Vaginal cervical swabs can be packaged together, while outer labia majora should be packaged separately, and inner labia minora, labia majora can be packaged together. Cartons should be labeled accordingly. Deposit evidence swabs into the package with the swab tips up and pointed in the same direction. This minimizes contact with the package and therefore potential sample loss. Ensure the swab packages are completely closed before placing them in the corresponding envelope. Seal the envelope with tamper-evident evidence tape. Then initial halfway on the tape and halfway on the envelope. Perianal swabs are collected around the anus for possible contact DNA, while anal swabs are collected for possible penetration. Please indicate on the collection envelope and swab carton if the collection is perianal or anal. One saline moistened swab can be used to collect all 5 fingernails of one hand, followed by the dry swab of each of the 5 fingernails. Both swabs can be packaged together in the carton. This would then be repeated with the other hand. Both swab cartons can be placed in the same envelope marked fingernail swabbings. Slides or smears do not need to be collected. The debris collection dried secretions envelope can be used for debris, such as rocks or vegetative material, the used condoms or swabs collected for possible contact DNA. Envelopes should be labeled with the patient name, date, and exact time of collection, the nurse's initials or signature, and the reason for the collection. Check the debris or dried secretions box. The outer envelope can be labeled with the description of the contents using the patient's words when applicable, as well as the location from which the items were collected. Use a patient's words to explain why a sample has been collected. Lab processes may vary depending on whether semen may be present or whether other contact DNA such as saliva may be present. This helps maximize the recovery of probative DNA by the lab. When collecting contact DNA swabs for examination, please include the patient's words on the swab carton to explain why the swab was collected. For example, "He licked my neck." Also include on the package the location the sample was taken. Dried debris should be collected in the folded paper that is included in the debris collection envelope. The paper fold must be secured with tape or a patient label. Seal the envelope with tamper-evident evidence tape, then initial the evidence tape halfway on the tape and halfway on the envelope. Clothing other than panties should be packaged separately. When panties are collected, label the outer sack box to indicate that the panties are contained within the box. Include the patient's name, medical record, date collected, time, collector's names or initials, and a description of the item. Place the panties in the undergarment bag. Fold the edge of the bag. Seal the bag closed with evidence tape and initial the bag halfway on the tape and halfway on the bag. On the bag, place a patient label, date collected, time, collector's name or initials, and description of the item. The chain of custody portion of the bag should be completed only when the items are packaged separately and will not be found in the sack box. When completing the sexual assault examination forensic report, it is important to press firmly when using the triplicate form. If known, consensual sex in the last 7 days should be noted, including the date, instead of just yes. This information helps the lab determine whether foreign DNA can be uploaded into the federal database or CODIS. If not otherwise noted on envelopes, the narrative section may be used by the lab to ascertain whether semen or non-semen DNA is possibly present. This can impact the type of analysis performed. The laboratory compares the information in the evidence items included in kit section against the actual contents to ensure no samples have been lost. Once all the items are collected and placed inside the box, seal the sack box with tamper-evident evidence tape and initial and date the tape, marking halfway on the tape and halfway on the box. Blood vials and urine containers from toxicology testing must be labeled with a minimum of 2 identifiers, including name, date of birth, and/or submitting agency case number. Alternatively, the hospital label containing the aforementioned identifiers can be used. The toxicology laboratory will reject any evidence that arrives without at least 2 unique identifiers. The blood vials need to be placed in protective wrapping sealed with adhesive tape and placed in a biohazard bag with the urine before being placed in an additional container. Specimen security seals need to be filled out with the date, time, and collector's signature. Make sure the labels placed over the blood and urine vials do not obscure either identifier. The urine container should be sealed with a leakage barrier, such as parafilm M laboratory film, as well as patient label or other tamper-evident evidence tape. Parafilm is not in the kit but can be generally found in a hospital for other uses. Once the evidence is secure, place it in protective wrapping sealed with adhesive tape. The urine container should also be placed inside of a plastic biohazard bag with the blood vials. The bag should be sealed by using the adhesive strip prior to being placed in the outer container. Once all items are collected and placed inside the box, seal the toxicology package with tamper-evident evidence tape, and initial and date the tape, marking halfway on the tape and halfway on the box. The outer box can be labeled with patient name, date of collection, attending examiner's name, and law enforcement agency case number. Alternatively, the hospital label can be used for identification purposes. The chain of custody information should be completed as appropriate. When writing on the outer box, please avoid pens with gel ink as it can wipe off or smudge. Identifiers must be consistent throughout all pieces of evidence.